Hello dear students, you must have come across through the checkpoints while studying the chapter cell cycle and cell division, right? What are they and how they are important to us? Let's understand with the help of an example. Suppose you are driving a car on a road. There are some signals which tell you whether to move forward or not. Exactly these checkpoints work. These checkpoints give a signal to a cell whether to move forward or not. There are various checkpoints. Major checkpoints are the G1S, G2M and the M checkpoint. What do they check for? G1S is the checkpoint which checks for the cell size, whether there is DNA damage or not. This checkpoint is the major checkpoint. It is also called the restriction checkpoint. Why? Because if a cell crosses this checkpoint, then a cell is committed to divide. So this is a major checkpoint. Then next checkpoint is the G2M checkpoint, which also checks for the DNA damage and whether the DNA replication has completed or not. And at the end, there is one more checkpoint called the M checkpoint, which checks for the arrangement of the chromosomes on the metaphasic plate. So let's understand them one by one in more detail. These checkpoints are controlled by regulators, which include the proteins like cyclins, enzymes like CDKs, and the complexes like APC, which stands for anaphase promoting complex. What are cyclins, CDKs, and the APC? Cyclins, as the name indicates, they form with every cycle. They are not constant. So, there are specific cyclins for every phase, like G1 cyclins, G1S cyclins, S cyclins, and the M cyclin. These cyclins, they do not work alone. These cyclins work with CDKs. What are these CDKs? Cyclin dependent kinases. So cyclins and CDKs, they form a complex. This complex is activated complex which phosphorylates the targeted protein, and then this targeted protein perform the function. So a lone CDK is inactive, but the binding of a cyclin activates it, making it a functional enzyme and allow it to modify the target proteins. This is a beautiful picture. With the help of it, we can understand more clearly. When there is no cyclin, then CDK is inactive. There will be no phosphorylation of the targeted proteins. If G1S cyclin is present in a cell, then this cyclin binds with the CDK, it forms the activated complex, then there will be phosphorylation of the targeted proteins, and then the S phase begins and the DNA replication enzymes are activated. So G1S checkpoint is supported or regulated by the G1S cyclins and the CDKs. If this complex is activated, then only DNA replication can be started. Now next is MPF, which is maturation promoting factor. What it is? When the cyclins bind to the CDKs, they form another complex called the MPF. MPF, also known as mitotic promoting factor or the mitosis promoting factor, it leads the cell or pushes the cell in the M phase. See here, if M cyclin is present in a cell, this combines with the CDK, then this complex, which is the activated complex now, it phosphorylates the targeted proteins, and then this helps in the spindle formation, chromosome condensation, and the breakdown of the nuclear membrane, which happens in the very first phase of the mitosis called the prophase, right? We can clearly see here that there is disintegration of the nuclear membrane which takes place in the prophase, and why it is so? Because it is due to the MPF, right? That is another checkpoint within the M phase when the cell is promoted from metaphase to anaphase. This is called the spindle assembly checkpoint. Here APC works, which is anaphase promoting complex. What does this APC do? This APC helps in the chromatid separation or the daughter chromosome separation. How? Let's understand with the help of a beautiful figure. This APC adds this ubiquitin protein on the securin protein. Why? Because to separate the chromatids, separase is required. A special protein or the enzyme is required called the separase. But this separase is tightly held by the securin protein. So if we want to separate the chromatids, we have to make separase free from securin. This APC complex adds the ubiquitin on the securin. So wherever this ubiquitin is added, that protein is degraded. Then what happens? The securin is degraded. Now separase becomes free. This separase, when becomes free, becomes active. This can lead to the chromatid separation. These two chromatids are held by the cohesion protein between them. So this cohesion is chopped up by the separase and then it leads to the separation of the chromatids. I hope you have understood the video very well. Thank you. Stay tuned with us.